lunch. Each of our presenters will talk for about 10 minutes, allowing for 5 minutes of questions. Remember to enter your questions into the Q&A part of the Zoom webinar. Next up is Aravind Patak, Associate Professor of Radiology, Oncology, and Biomedical and Electrical Engineering at the Johns Hopkins Kimmel Cancer Center, the Institute for Nanobiotechnology, and the Institute for Computational Medicine.
Thank you, Dr. Berger. Can everyone hear me? Let me just...
Yes, I can hear you. Yeah.
Okay, great. Let me just share my screen here real quick. All right, so good morning, everyone. And it's really fantastic to be able to join you and follow all these really exciting presentations with some of the work that we've been doing on cancer and how we're leveraging all kinds of cool technologies and techniques to gain insights into not just cancer biology, but to develop and guide precision medicine therapies that have been the theme of this morning's talk. So what I'd like to do is just start with a basic background so that we're all on the same page. So cancer biology, just like most of biology, when we're trying to interrogate it in our lab, it's usually more than the sum of its parts. And the reason for that is very straightforward, is because you have all kinds of different tissue components interacting with each other in all kinds of unpredictable ways. And often, even though we'd like to zoom in on certain aspects of the biology that we're studying, much to our detriment, there are a lot of complicated factors that kind of impinge upon what we're trying to study. And what I mean by that is there's a lot of inherent complexity in the underlying tumor biology or the biological system we're trying to look at. And the reason for that is because we have many different cell types that are often interacting with each other. There are other tissue components like blood vessels and supporting tissue that also interact. The tissues and the cell types tend to be very heterogeneous. They're dynamic systems, so they tend to change over space and time. They tend to be what are known as nonlinear systems, so they don't change in predictable, straightforward ways, so we need to keep that in mind. And then, like I was saying, these different components kind of feed back and inform each other. So it's not always as straightforward as it looks. So what we try to do is we try to reduce some of this complexity by turning to model systems. But when we do that in our labs, we need to bear in mind that these systems need to be realistic. They need to mimic what we see in a patient or in nature. They need to mimic the dynamic nature of these systems, and they need to be high fidelity. So they need to be able to reproduce the pathology that we're seeing with very high accuracy and precision. And often these biological systems in our labs, they require to be tested very rigorously, these model systems. We need to validate them to make sure that they're behaving like we expect. And then we need to be able to perturb them either with a drug or with some kind of therapy that kind of mimics what we'd expect to see in a patient population. And since we can't experiment on people, we tend to do these in two very large spaces. We work in what's called the preclinical space, so a various range of animal models, whether it's cancer or neuroscience or heart disease, or we work in the in silico space, which is basically where we use computer models that are informed by certain physiological rules. So our lab kind of straddles a very interesting space. So we are a group of interdisciplinary scientists with backgrounds ranging from electrical engineering to biophysics to computer science, and even art is applied to medicine. And we work in 4 big areas. So we developed new imaging methods. So a lot of you might be familiar with MRI, ultrasound, and CT. So we develop a lot of these methods in the preclinical space before they actually enter the clinic. We develop completely new technologies which allow us to answer some of the biological questions in this preclinical space. We develop new computer-based tools to model some of the phenomena that we see in patients. And then we come up with really cool data visualizations, because as you've heard in a lot of these talks, as technology advances, we are generating gigabytes, terabytes of data that need to be handled and visualized in very unique ways. So we kind of sit at the interface of these disciplines. And I'm going to show you examples of each of these to kind of drive home the point of what I mean by them. So shown here is an MRI scan from a patient with a low-grade brain tumor. And very often, these patients come into our clinics, they undergo what's called a pre-surgical workup, where we image their brain and we map out what are known as eloquent parts of their cortex. So what I mean by that is we map out parts of their brain that are important for really basic functions like speech, respiration, and things of that nature, so that when a surgeon comes in and resects the tumor, he takes care to stay away from those eloquent regions of the brain. So this patient was asked to do a very simple task. He was asked to move his tongue in the MRI scanner. And then we are able to map out the parts of the brain that control the tongue movement. So these yellow areas actually show that for different slices in the brain. But then you also see something very interesting happening, is that the part of the brain which has the brain tumor, you don't see any pixels, if you will, light up. And for a surgeon or a neurosurgeon, it's kind of important to know why that's happening. So if you zoom in into a pixel, you can actually see what this cartoon shows here is what's happening inside a pixel. So this is what's known as a neurovascular unit. And there are hundreds of thousands of these inside one imaging pixel. And what this shows you is a schematic of a neuron here. So the patient's asked to do a task, the neurons fire. Once the neurons fire, they communicate to the nearest blood vessels via what are known as astrocytes. It's just a different cell type with these little feet that impinge on these blood vessels. And the astrocytes kind of signal the blood vessels to dilate and deliver more blood to that area. And that blood is what we image in an MRI scanner. So it's really important to understand when we have cancer cells present in this little unit, how they perturb this signaling, because it can actually not just mess up the imaging that we're seeing, but it can actually give us some really important information regarding the disease progression. And it's very hard to answer these questions in the clinic. So we actually turn to the preclinical space to do that, to see what happens when cancer cells are present in a patient with brain cancer. So this is the exact same imaging experiment, if you will, done on a mouse. So this is a mouse cross-section here. This is the brain of a mouse, and the color-coded areas are different brain regions. These are the lungs and the esophagus. And you can actually see that in a healthy mouse, these two brain regions communicate with each other using this fMRI method, functional magnetic resonance imaging method, very nicely. But then what we can do is introduce a tumor into the mouse's brain, shown here in red. And immediately you can see a couple of things happen. One, the structure of the brain is dramatically altered. And two, the communication between these two brain regions is dramatically altered. So like I told you before, if you look at the neurovascular unit, the question is what's happening at the level of these cells here? So if you look at sections from this mouse brain, so these are images from the brain of this mouse after it's been euthanized, and the tissue is color-coded or labeled with things that recognize these different tissue components. So shown in green are the astrocytes, shown in red are the blood vessels, and shown in blue are just the cells. So you can see this is a healthy brain, and this is the boundary of the tumor, and this is the tumor brain. And if you zoom in, you can see healthy blood vessels are very nicely surrounded by these astrocytic end feet. But if you look inside the tumor, you see an absence of these astrocytic end feet. So the idea is to use the preclinical space to try and understand some of the things that we're seeing in the clinical space and get a better idea of what's happening in brain cancer patients, both from the standpoint of therapy, disease progression, as well as from the standpoint of creating new clinical biomarkers. So the other thing that we do is develop new technologies, because although MRI is a cool technique, if we wanted to follow the disease every single day, hour by hour, even in a mouse or an animal model, that's kind of really challenging to do. So what we also do is we develop our own imaging instruments. So we develop what we dub a GoPro for a mouse. What that does is basically is a specialized camera that we built ourselves in-house that sits on a mouse that has got a cranial window, which allows us to peer into its brain continuously over the entire lifetime of the mouse or the disease we're studying. And this is a very special camera that allows us to take pictures of different kinds. So we can actually inject a fluorescent tag, and we can have the blood vessels light up, and it'll show you here in this movie. You see the veins and the arteries light up, just like I was telling you earlier. You can actually see the neurons fire when the mouse is performing some kind of task. In this case, the mouse was asked to listen to some kind of tone, and all these neurons fire. And then you can see this blush of incoming blood flow to supply the neurons that just fired. So this is a very, very powerful tool that we created in our laboratory to actually interrogate the brains of awake, freely behaving animals, so you don't have the confounding effects of anesthetics when you're studying the brain. But the real power of the technique comes through when you're looking at something like brain cancer. So what we did was we imaged an animal with a brain tumor over the entire lifetime of the tumor before the animal succumbed to the tumor. So shown here on the top row are basically images taken through this window using the specialized camera, where we have brain cancer cells that are labeled with a green dye. So as you see, as the brain tumor kind of progresses, the amount of green in your field of view increases, showing you that the tumor is expanding right under the camera. But then if you take a picture with this camera in another channel, you can actually see that there's a little injury where we put the cells. And then you can see blood vessels come in a process known as angiogenesis, which literally means the birth of new blood vessels. And they come in and sustain the growth of the tumor over the entire lifetime of the tumor. And not only are these blood vessels growing, but they actually start to become functional, because these are now images of the blood flow in these tumors. So you can actually see blood coming in and flowing. So again, this is a very, very powerful tool that we and other people are now using, these cameras that we've made, to answer chronic questions about disease, whether it's brain cancer or stroke or other neuropathologies. We now have a tool that allows us to see the effects of therapies, progression, and so on over the entire lifetime of the disease model, which would have been very tough to do with any other kind of imaging method. The other thing that we do are we develop new computer-based tools. So one of the things in cancer that's been a really important problem is this issue of heterogeneity. So what I mean by that is that the same cancer presents very differently in different types, in different patients. So that's what we call interpatient or population level heterogeneity. The same tumor type, when it metastasizes or spreads to the different parts of a patient's body, can present very differently, both at the genetic and the phenotypic level. So that's what we call intrapatient heterogeneity. Sometimes the same tumor looks very different depending upon which part of the tumor you biopsy or sample. So that's what we call intratumor heterogeneity. And finally, of course, at the genetic level, cells are very different. So we get the genetic level heterogeneity. Since we can't repeatedly biopsy patients because they're very sick, it's an invasive procedure, it's also prohibitive to do large cohorts of animal studies, we often turn to computer-based approaches where we can actually develop new tools to interrogate and develop kind of computer models that allow us to get a handle on this heterogeneity. So you saw this gorgeous talk earlier on how computer models are used to look at cardiac arrhythmias. So we do something similar. We create an atlas of tumors developed in animal models. So shown here are MRI images. So the soft tissue is shown in gray. So that's a tumor, a breast tumor. In red, you actually see the blood vessels that actually supply the tumor. They're color-coded by their average vessel radius. And these 3 samples mimic an early stage breast tumor, and these 5 samples mimic an advanced stage breast tumor. So once we have these digitized atlases, we can kind of take a Google Maps type of approach, if you will, to simulate, using some mathematical rules, what's actually happening inside these tumors. So using the Google Maps analogy, all the blood vessels are kind of like the streets in a Google map. The traffic on those streets is the blood flow. And then from that, we can actually infer other parameters in these image-based models, like I'll show you. So shown here is a map of about 100,000 blood vessel segments. Once we know what the blood vessel architecture is, we can actually map geometric properties. In this case, it's a tortuosity, so how tenuous or how circuitous a vessel is. We can actually simulate what the blood flow velocity is in these tumors. We can compute what the average oxygenation is in these tumors, in these 3D tumor maps. And we can actually start to now infer what drug delivery would look like, what therapeutic response would look like, and other things that we were interested in the patient population. So hopefully with these examples, I've been able to show you what we can actually do in these spaces with these tools, because these tools actually inform the clinical workflow as well. So I'd like to end there, thank the collaborators that work with me on this project, some of our funding agencies, and of course, the incredible students that have done this work. Happy to take any questions.
Great, thank you very much, Arvind. So we have one question from Stephanie Baum, who asks, when you spoke of mapping out the eloquent portions of the brain to assist surgeons so they avoid these areas, Do these locations vary widely between patients?
Yes, they do. They do vary widely between patients, and we have certain ways to take that variation into account. Often, the data that we see is mapped back into a standard space. So we have atlases of the brain where we can actually map everything back to a standard set of coordinates to reduce some of that variability. But also we do scans on a patient by patient basis. And also when patients have a brain tumor, the brain will often move, or due to surgery or treatment, there will be shifts in the location and the spatial kind of extent of those tumors. So we have very patient specific information. So we are doing precision medicine for these patients actually already in our workflows. But now we're trying to get even more precise using some of these tools.
Wonderful. Thank you again.